So I'm Heidi Cook-Andersen. I'm also in the REI division, and all the animation is already there. So anyway, yeah, it's there. Yeah, so the long-term goal of research in my laboratory is to understand the mechanisms required to drive the transition from this fully differentiated oocyte to the totipotent embryo that can then drive development of an entirely new organism. And one thing that I find really fascinating about this transition is that it happens in the absence of transcription. So transcription is globally silenced here in the oocyte just before ovulation, and it doesn't fully resume until the late 2-cell stage here in the mouse, and even later at the 4 to 8 cell stage in the human embryo. But without transcription, this means that all of these critical developmental events in between, including ovulation, oocyte maturation, fertilization, and even reprogramming to totipotency, are dependent completely upon post-transcriptional mechanisms. And we know at least one important way that this occurs is through unique pathways in the oocyte that regulate gene expression at the level of translation. So this was supposed to show is that during oocyte growth, when transcription is active, the oocyte is able to accumulate a large body of mRNA. And some of this it uses immediately, but a lot of it will store in an unusually stable state for use at a later stage. And then once transcription is arrested, the oocyte can then access this mRNA pool and use translation to turn on and off gene expression. And it somehow knows to activate translation for specific sets of mRNAs that are needed at each stage of development across this transition. And equally striking, I think, is that almost all of these oocyte-contributed mRNAs are essentially degraded before new transcription begins in the embryo. And this is really, we think, the basis of life from flies to humans, but something that we still understand surprisingly little about. So this is the goal of my lab, to understand the mechanisms that really drive this transition and the post-transcriptional mechanisms in particular. And all the projects in the lab are aimed at understanding like different points of this transition. So for instance, we're asking how transcription can be globally silenced in the oocyte, and we have a really strong start on this. So a couple people in my lab, Jennifer Dumdai and Kyuto Chol, have recently published findings in Developmental Cell that show that RNA decay plays a critical and surprising role in this process. And then kind of looking at the other end of this transition, we're interested in understanding how the processes that lead to global transcriptional silencing are reversed to allow transcription reactivation in this 2-cell embryo after days of transcriptional silence. And our favorite hypothesis here is that perhaps the very promoters for the very first genes are somehow uniquely marked, and that transcription is kind of set up well in advance, perhaps even in the oocyte before transcriptional silencing occurs. And then the other major project is really focusing on events here in the middle, trying to understand this activation and inactivation and translation and decay that really that allows this transition to happen without transcription. So it's been challenging, but we're working to address this question in mammals. And we're doing this not because we're hoping that our results will be relevant not only to advance our understanding of development, but that it'll also help us improve protocols to improve the efficiency of cell reprogramming in vitro and to improve diagnosis and treatment of infertility. And to address these challenges, our lab, together with our collaborators, are working to adapt and develop novel techniques that allow us to address these questions using a very small number of cells. We're very excited because these new protocols are giving us a unique opportunity in the field to address these questions in ways that we haven't been able to before. Thank you.